Let me see if I can get myself to show. Hmm, I don't know why this is not showing me. This is something new. I'll have to take it into Clark College, I'm guessing, because I'm not able to show myself or show you guys anything like at my desk. I'm not sure what's going on. This used to work, and
Under the settings, have you checked to make sure that your I have a green screen isn't toggled on?
I have it on, but I've got a green screen behind me.
Oh, gotcha. Okay.
Like I said, I don't know what the problem is. I'm gonna have to go into Clark College because I didn't change anything. It must have been an update in the oh, what do you call it? The program for Zoom. All right, let me share my screen. Are there any questions before we begin?
Actually, I did have a quick question regarding I believe it's the worksheet post we're all meant to answer one of the 21, 22 questions on.
Yeah,
Um, when all the questions have been answered and you wish the rest of us to either go back and check to see if any of those questions need to be revised, if we can add anything to them, would you prefer we create our own post regarding that question number or to reply to the individual on their post when they initially answered the question?
you should apply a new post.
Okay, create a brand new post and
Yeah,
just
create a brand
reference
new
the
post.
question number.
Yeah. So that
Okay,
way people know where you are. And you're supposed to take the questions in the order they are in the worksheet. That way it's not confusing as to which question has been answered and which needs to be answered. And I understand that students have not been doing that. The first person who did not do that will get a penalty, but because so many people have been taking the questions out of order, only the first person will get the penalty. Now, you need to follow the questions in the order of the worksheet. And if you don't like that question, all you need to do is don't answer it and come back to the worksheet at a later time and somebody else will answer that question. Okay?
okay. Yep.
So always take the questions in the order of the worksheet. And if all the questions have been taken, which obviously will be the case today because we have a lot more students than 21 questions, um, you just find a question that you think is wrong and give a correct answer. Or if all the questions are correct, you can just start the worksheet over again, meaning start with question number one, and then the next student will take question number two. Is that clear? So if you do not follow the worksheet order, it'll be very difficult to figure out which questions have been answered and which questions have not been answered. So you must follow the worksheet order. And normally I will give a penalty for everyone who does not follow the worksheet order. However, my understanding is that a whole bunch of people have not been doing it, in which case I won't apply the penalty to everyone because obviously people have I don't know, been confused because of the way this worksheet was done. So you guys are supposed to follow the worksheet questions in the order of the questions in the worksheet. Any question about any of that?
Yes, I just have one question. I also emailed you, but so for me personally, I saw that number, what was it? I think it was 12 through 19 done. So I decided to do question 20 because that was in the row for me. Will I get penalized for that? Because it looks like someone missed, I think, question 9.
You will not be penalized if you skip question 9 and a whole bunch of other people skip question 9. Is that what you're asking?
Yeah, so someone, instead of just putting question 9, they put question 19. That's what they chose. At least that's what it shows on my canvas. And then
I don't know. So just go on.
and then people started off from like 10 to like 19 is what I saw. And so then I just answered 20, even though 19 was posted after 8 and
Okay,
I
yeah, you
posted
shouldn't have
after.
done 19. If somebody went out of order, you should have got back to the correct order. Okay,
Okay,
so
so
in
you
the
would have.
future, go back to the correct order. Like I said, I'm only going to penalize the first person who um, took the questions out of order.
Okay,
I
perfect.
changed the way this works too. I'm trying to find where we left off, and I'm not finding the. There it is, find and replace. All right, I've got it marked by 2 here. Hopefully you can see my screen. Let me state that I do need to make an announcement, and that is next week I've got a report. Uh, not really report, but I got to call in and see if they requested my presence for jury duty. Meaning next week I may have jury duty. I'm not sure what time I will be able to get home, and I may not know until after I've done it, actually. I'm not sure I'll be able to get home by 5 o'clock, in which case this class the lecture may be online. I will not know probably until Monday if I'm going to be serving jury duty on Tuesday. And I may not know really until Tuesday, but I will make an announcement. And like I said, if I can, I will run this class in the normal fashion with Zoom. But if I cannot get home by 5 o'clock, then this class will be online if I am selected for jury duty. And I tried to get out of it, and they didn't let me at least do it online. Maybe I can get out of it if I go speak to a judge. But, and the other thing is, if I can get home by 5, this class will not be interrupted. But I don't know what time I can get home by. And it's possible I may not ever be selected for jury duty as well. I'm just warning you that next Tuesday I may be in jury duty, in which case this class may not be meeting at the normal time. I'll have to give an announcement, and you may not get that announcement until Tuesday. Hopefully you can get it by Monday. I'm not sure when I'll know. Any questions about any of that? All right, I think the 6:30 lab will run normally. I'm not sure if any of my other classes will run normally. In fact, I'm going to guess that if I'm selected for jury duty, it will definitely impact the 2:30 lab. I may not be selected for jury duty, but I might be, and I can't see if I'm selected for jury duty it not impacting the 2:30 lab, in which case I'll just have to post a video and it'll be a totally online class for as long as I'm in the jury duty, which lasts for either a week or 2 weeks, whatever the case is. Any questions about any of that? All right. Hopefully it won't impact this class, but it might. And like I said, I will make an announcement when as early as I know if I am selected for jury duty. The question is, I don't know when I'm going to know, and it may not be until as late as Tuesday. Hopefully, I'll know at least by Monday evening. All right. Any questions about anything else before we begin? Let me go to the syllabus. So today we're going to be continuing Chapter 1 and Chapter 10. And that's really it for the lecture. So I have that we were on this slide last time, and we did mention that microorganisms and microbes are nearly the same thing. The difference is I consider a virus and a prion is not a microorganism, but they are a microbe. And other authors do use these two words synonymously, like I think your textbook author does.
I hate to interrupt, but your screen is not sharing right now.
You got to be kidding me. I
No,
got to
I'm not.
stop share on. All right,
Yeah,
let's try
yeah,
this.
it's not showing that you're sharing, and we
I
can't.
had the share on, okay? Let me go ahead and show. Is it showing now?
It is. Thank you.
Yeah, I don't know why that wasn't showing the first time. All right. And you do not see me, is that correct?
Correct. We don't see you,
Yeah,
just your screen.
I'm not sure what's going on there. I'll have to take it into Clark College because I've got the green screen now and still the screen. I'm not showing. Um, it's nice when I can show, like especially in the lab, when I'm trying to demonstrate something and then I can show it to you. All right, let's go on. So viruses are acellular, meaning they do not have cells. They do consist of DNA or RNA as their core innermost molecule, which is their genome. And it will be either DNA or RNA, but never both. This core molecule is surrounded by a protein coat. The coat may be enclosed in a lipid envelope. And when do viruses replicate? They only replicate when a virus has infected its host cell. Viruses are not part of the three-domain system of life because they're not cellular, and it's debatable whether they're alive or not. We're not going to go into that debate. When we're talking about the three domains of cellular organisms, they are the bacteria, the archaea or the archaebacteria, and the eukaryotes. When we look at their cell types, the bacteria and the archaea both have prokaryote cell types. The eukaryotes have a eukaryote cell type. When we look at their chromosome or their DNA, the bacteria generally has one chromosome. It's a circular molecule, meaning it ends where it begins. Bacteria have no histones, no introns. Eukaryotes, on the other hand, have many chromosomes. They're linear, like when we look at the X chromosome over here. It has a beginning place and an ending place. It has histone proteins in the DNA, and it also has introns. We'll talk about them later. The archaea are something in between the bacteria and the eukaryotes. You don't really need to know that for now, but they generally have one circular molecule. The difference is they do have histones on the DNA. When we look at the ribosomes, the bacteria and the archaea have smaller ribosomes than the eukaryotes. The eukaryote ribosomes is 80S in size. The archaea and the bacteria have 70S size of ribosomes. The size refers to the sedimentation coefficient or where the ribosome sediments in an ultracentrifuge gradient. You don't need to know that, but you should know that the bacteria have smaller ribosomes in the eukaryotes. It's 70S versus 80S. When we look at the cell wall, the bacteria, if they have a cell wall, have peptidoglycan in the cell wall. The archaea and the eukaryotes have no peptidoglycan in their cells. So that's another difference between them. Any question about any of that? All right, we're going to change gear a little bit and talk about the history of microbiology. We're going to have only talk about a brief history of microbiology, which largely concern the debate over whether organisms could spontaneously generate or whether they came about from another living organism, and that became known as the theory of biogenesis. The history of microbiology also encompasses the golden age of microbiology, where wherever a microbiologist looked, they essentially discovered something new. And that happened, you don't need to know the years, but from 1857 to about 1914. That's the golden age of microbiology. Any question about any of this? All right, there's a brief history of microbiology. In one slide, we'll talk about some of these events. And this is the golden age of microbiology, shown in golden. The history of microbiology begins in ancient times. The only date you need to know in the slides of the history of microbiology are that greater than 3.5 billion years ago, we can find fossil records of the ancestors of bacteria, and they were the first life on the planet Earth. There's a fossil record of an early life form. You could say the ancestor of bacteria, but really you could say the ancestor of archaea as well as the ancestor of eukarya. And there's an artist's rendition of it. And there's some more. That's the only date you need to know in the history of microbiology. I'm going to mention the other dates just to put them in chronological order, and it's a little easier talking about them. But the next date that's important in microbiology, or the next event that's important in microbiology, is the early Babylonians wrote on their clay tablets recipes for making beer. And if you don't know, beer was made by microbes, which the Babylonians did not know, but they did make recipes for making beer, and that happened 43,000 BC. The next date I'm going to mention is 600 AD, where the Mayans recorded making a fermented beverage from cacao that we would call chocolate. And really, the only thing you need to know is, well, you do need to know the date here, but what the event was and why it's important to microbiology, meaning this is the first record we have of people using microbes to make beer, and then another record we have of microbes used to make chocolate. And I guess you should also know the event that before chocolate, we could write about chocolate being made. Someone had to write about beer being made. And even before that, you had to have the first life show up on the planet. So you kind of need to know the order of things here. The next important observation in microbiology was in 1590. You don't need to know the date, but you should know that Janssen developed the first microscope. It was a compound microscope. And obviously it was important for microbiology because we couldn't see microbes until the microscope was invented. However, the microscope was never used to look at life until 1665, when Robert Hooke reported that living things were composed of little boxes that he called cells. Let me get something Hopefully I can get paint to come up. And they changed this, so I think I've got to do it twice before paint will come up. There we go. What he did was he took a look at shavings of cork and noticed little boxes in them that he called cells. They need to be square. I'm not drawing them perfectly square, but you're getting the picture. Shouldn't have made this so long because it's going to take me a while to get there. Make it a bigger box. There we go. They were called cells because in a monastery, there's the monastery, the monks would walk down a hallway and then they would have a little room that they called cells. Obviously, they had a door to get into the room, and you don't see a door on in a cell, but it looked like this. And he thought these boxes of that he saw on the shavings or the cork looked like the cells of a monastery. And so that's where the term cell comes from. Any question about that? All right. No question about that. Let me end that. Now, Robert Hooke noticed the cells in plants when he looked at plants, and obviously cork. He looked at first saw these cells in thin shavings of cork under the microscope, which is the bark of the cork tree. And we're not going to talk about cork, but it's actually used in three ways. You call it a cork when you plug a piece of cork into a bottle of wine, that we call a cork, and you remove it to drink the wine. And the cork comes from the cork tree, and the bark itself is also called cork on the cork tree. Any question about any of that? Anyways, in Robert Hooke, whenever he looked at plants, he could find cells. But when he looked at animals, he did not see cells. Now, the primitive microscopes he was using tended to blur the vision of things you were seeing. And what do plant cells have and animal cells lack that made the plant cell show up in Robert Hooke's primitive microscope, but animal cells did not show up in his primitive microscope because they were lacking something in their cell? What was it that animal cells lack that plant cells have? Come on, somebody guess.
Cell
No.
walls.
Yeah, cell wall. Plant cells have a cell wall and it's very thick and very rigid. And the primitive microscope could see that cell wall in the plant cell, but animal cells lack a cell wall. They only have a cell membrane. And the primitive microscopes could not focus well enough to see the cell membrane. So initially, people did not see cells in animal tissue, but they did in plant cells. But anyways, his writing of cells began what became later known as the cell theory, that all living things were composed of cells. It was developed by others besides Robert Hooke, and it was actually written up formally and published by Virchow. The above, everything I mentioned, is not specifically microbiology. The discovering of the microscope isn't really doing anything about microbiology. And then Robert Hooke never looked at microbes. He was looking at plant and animal tissue. So he wasn't really looking at microbes when he saw cells. Now, remember that I mentioned that the history of microbiology was largely concerned about how life originates. In the 1800s, you don't need to know the years, just the early microbiology, there were two competing hypotheses to explain how life began. One was that life began spontaneously from inorganic matter. A vital force moved into the inorganic matter, creating life. And that was known as the theory of spontaneous generation. The other theory was biogenesis, and that is all living things came from another living thing, the theory of biogenesis. Nobody ever argued that a baby ever spontaneously generated, but simpler life forms, they argued, could spontaneously generate. Any question about any of that? So let me just state that even in the very beginning, people always said a baby came from the two human parents. But over a century ago, beliefs included things like if you threw a log into the fire, sometimes a newt would crawl out. And people who believed in spontaneous generation said, spontaneous generation of the newt. You put the log into the fire, and the fire and the wood of the log created the newt, spontaneous generation. And then people would note flies emerging from manure, and they would say, the vital force went into the manure, giving rise to the flies. And then the same thing they would say about maggots arising from a decaying corpse. The vital force went in, and then the maggot formed. Most were logical, not any were scientific. Some were even amusing. Like someone wrote and published that if you put sweaty underwear in a jar of wheat, it could spontaneously give rise to mice. In reality, the mice crawled into the jar to get the wheat, and then they got stuck in the jar, and the sweaty underwear had nothing to do with it. Any question about that? All right, just a brief sidetrack. Microorganisms, when they were growing in media, in a broth, they would make the broth become turbid, sort of like this. Let me blow that up a little. So that would be a clear media, like when you first make the media. And if you let it sit out, it would become turbid like that. And there'd be swimming microorganisms all in this broth. And we often saw this initially whenever people made media, it almost always would become turbid. The first evidence proving or disproving the theory of spontaneous generation came by Francesco Redi, who actually you should also know, besides being the first person to give evidence against the theory of spontaneous generation, he also was the first person to write up a controlled scientific experiment. So he's important in science as well as in spontaneous generation. What he did was he made 3 jars that he left open, and he put in slices of meat, and maggots appeared in the meat. And then he had 3 jars that he completely sealed, and they had no maggots. And the people who argued for spontaneous generation said, no, Francesco, you completely sealed the jar, so the vital force could not get into the meat and then give rise spontaneously to the maggots. So he ingeniously covered the jar, put in meat, covered the jar with a fine wire net. And he found in these open jars covered by a fine wire net, it was a metal wire, that they had no maggots in the meat. And this did not support the results of spontaneous generation. And after Francesco Redi's experiment, nobody believed that complex life forms could spontaneously generate. Because the theory, the evidence of Francesco Redi said that flies could not, or maggots could not arise spontaneously in the meat. However, microbiologists would argue that microbes could grow in this meat. Obviously, that wasn't known in 1668, but later on, people were able to observe microbes growing in the meat. And so people argued for at least a century after Francesco Redi's experiment that microbes could spontaneously generate. And like I said, whenever people made media, we usually saw microbes growing in the media some point later in time. About this time came the first truly observations of microbes. In the 17th, 18th centuries, this guy here, Antony van Leeuwenhoek, from 1673 to 1723, observed the first microbes. He found microbes wherever he looked, microorganisms and scrapings from his teeth, and rainwater that he collected, although the rainwater had to sit a while, and peppercorn infusions. He found lots of microbes, tons of microbes in water collected from the Thames River. And the people of London didn't like hearing that because they were drinking that water for their drinking water. And there were tons of microbes in it. So wherever he looked, he found microbes. And there he is there. The reason why he was able to find lots of microbes essentially wherever he looked was because he was the best lens grinder of his day and also the best microscope builder. This is an actual microscope that he built, and you'll note that it's a simple microscope containing only one lens, so it's really nothing more than a super duper magnifying lens. But he actually ground that lens himself and then built the microscope stone here. And the pen, that's what he stuck in whatever he was observing under the lens. And then how this works is you just hold it up to a light source and look through the microscope, which you're actually seeing right here. I have to blow it up. There he's using a light from the window, the sun, to observe his whatever he's looking at under this microscope. And there's a simple microscope there. Any question about any of that? He was so good of a lens grinder and microscope builder that he saw cells so small that nobody else saw them for about a century later. And here are his drawings right here of those cells I'm talking about. And if you don't know, that looks like a bacillus, that looks like a spirillum. These in E look like cocci, and these are more bacilli. He actually saw and drew bacteria, cells that were so small no one else could see them. And that's because they didn't have his fabulous microscopes that could focus in on them. It took 100 years after Antony van Leeuwenhoek published these results that the microscopes gradually got better. And after 100 years, the focusing got good enough that other people were finally able to see bacteria. Any question about any of that? And that's part of the thing about a simple microscope. It only has one lens, and so its focusing was clearer than a compound microscope because two lenses just were more difficult to focus. All right, after that, Rudolf Virchow proposed the cell theory that living cells can only arise from pre-existing living cells, and this became known as the concept of biogenesis. But arguments about spontaneous generation continued for a few years after Rudolf Virchow published that he thought all living cells came from only pre-existing other living cells. What happened in 1861 was Louis Pasteur came on the scene and he provided evidence that disproved the theory of spontaneous generation, thereby confirming the theory of biogenesis. Hopefully I said that right. He disproved spontaneous generation, thereby confirming the theory of biogenesis. What he did was he demonstrated that there are microorganisms in the air. And if you make media or something, and then microbes fall out of the air, it would grow in even a sterile tube of media if the media was left open to the air. But he showed that microbes can be destroyed by heat. And then he made his ingenious, famous swan neck flask experiment, where he had a beaker with a very long neck. He put the media in. He then bent the neck of the flask by heating it, bending it like that, and then heating the media to kill off the microbes that were in the media to begin with, because they would be in both the media as well as falling in from the air. And then he let this sit, and the media never became contaminated. Furthermore, he showed that the media never become cloudy, turbid with microbes unless he poured the media down to here and then poured the media back. And what he proposed was is that microbes from the air, meaning this tube is left open to the air, so the vital forces could come in. But he showed that microbes from the air could drop down the tube, but then they'd settle there. They would not come up here because of gravity. And so to get the microbes to grow, you had to take the media down to here and then bring it back. So if the media was just here and the microbes were just there, no microbes grew here, showing that the theory of spontaneous generation did not occur because the vital force could get in, come to the organic matter of the media, and it never happened. Any question about any of that? If you ever go by the microbiology lab, for over 10 years, we've had a flask on the window of the microbiology lab like this that has been open for 10 years and has never had microbes growing in the media. Eventually, all of this media will evaporate, and I don't know, maybe somebody will do this again. I don't know. But it's been over probably 10 years now. So it's probably about 12, 13 years now. All right, let's talk a little bit more about the golden age of microbiology. It generally is said to start with beginning with Pasteur's work. Discoveries included the relationship between microbes and disease. This became known as the germ theory of disease. Out of this work came immunity, how a person could become immune to a disease. And then out of this work also came antimicrobial drugs, work on the first antimicrobial drugs that led to, among other things, but antibiotics. Some miscellaneous events. In 1850, Pasteur developed the basic idea of aseptic technique, so that if you had a culture that was pure one species, you could work with it using aseptic technique and then not contaminate that pure species. A little before that, maybe about a year before that, Pasteur developed a way of getting a pure culture. We'll talk about that in a lab in a future event. In 1881, Koch developed the technique for streaking for isolation on solid media. This was an easy way to obtain a pure culture, much easier than Pasteur's way. What he did was limiting dilution of a liquid culture, and he would have to make 10 replicas of that. And if only one tube had growth in it, and 9 tubes had no growth in it, Louis Pasteur correctly assumed that the one tube with growth was started primarily from one cell, and so that was likely to be a pure culture. Robert Koch developed it much easier, and what he did was he took bacteria smear or microbes, made a smear on a solid agar plate, and if he got an isolated colony, he would take cells from that isolated colony, streak it out for colony isolation again, and then take cells from that second isolated colony to grow up his pure culture. And even then, 100% of the colonies was not pure, meaning formed from more than one species of bacteria, but over 95% of the cultures were pure. In microbiology, it'd be very hard to get a culture that would be 100% certain would be pure. But if you streak it out 2 times, going through 2 colony isolation events, and then take the cells from that second colony to grow up your culture, you will have a 95% or higher chance of that being a pure culture. Any question about any of that? And that's such a good technique of getting a pure culture, we still use it today. Haas, about this time, developed nutrient agar, a new solid medium for growing mini microbial growth. Haas was actually a lab assistant of Dr. Koch, so he worked in Dr. Koch's lab. And then, similar time, Sergei Winogradsky developed the idea of microorganisms being involved in biogeochemical cycles, like the nitrogen cycle, the sulfur cycle, and we've talked about the nitrogen cycle. Sergei Winogradsky was able to discover that microbes were running the nitrogen cycle because Sergei Winogradsky did something that no other early microbiologist did, at least none that was published, and that is he tended to study microbes in a mixed culture where there are many different species together. Most people studied microbes in a pure culture. And the reason for doing that is if you have a tube and growing microbes in it, it turns yellow, then you know that the turning yellow happened by that one species you were studying because that's the only bacteria in the tube. However, if you're looking at a mixed culture with many different species in there and the tube turns yellow, you don't know which species turned the tube yellow and which did not. So generally in microbiology, we do study microbes in a pure culture, but to study something like the nitrogen cycle, you had to use a mixed culture of many different bacteria species to be able to see the nitrogen cycle. Any question about any of that? All right, about 1857, Pasteur showed that microbes are responsible for fermentation. Fermentation is the changing of the sugars in beer or wine, changing that sugar into alcohol. So Louis Pasteur showed that alcoholic fermentation is the conversion of sugar to alcohol to make beer and wine using microbes. He also showed that microbes are responsible for spoilage of food what he did was he looked at spoiled beer initially and discovered that other microbes were digesting the alcohol in the beer to make acetic acid, making the beer acidic and taste bad, and thereby go bad. Any questions about any of that? So Louis Pasteur showed both fermentation and then food spoilage happening by microbes. Louis Pasteur then showed that the spoilage bacteria or microbes growing in the beer or the wine could be killed by heat that was not hot enough to evaporate the alcohol in the wine. And this has been called pasteurization, the heating of a substance, usually liquid, but you could use it on gas, to kill off the microbes that contaminate the food and may cause human disease, meaning human pathogens. And this pasteurization does not kill all the microbes. It just reduces their number. Pasteurization is used in a wide variety of foods, primarily dairy products like milk. And if you buy pasteurized milk, it does have a shelf life a little bit longer than raw milk, but both of them have a shelf life. And the reason why pasteurized milk has a shelf life is that microbes are in it. Not all the microbes are killed by pasteurization. And although the human pathogens are all killed, there are other microbes in the milk that, given enough time, will grow, causing the milk to eventually spoil. Any question about any of that? And Louis Pasteur first used pasteurization on beer. This is just for your amusement. It was then widely applied to many different foods. And milk is the number one thing we use it on today. Although I guess most beer is pasteurized. So it's probably the number two thing it's used on. With the concept of fermentation, that microbes can affect organic matter, changing the sugar in the beer or the wine into alcohol. And then the idea that other microbes could grow in the beer or wine, digesting the alcohol and making acetic acid, spoiling the food. All of this led to the concept that maybe microbes have similar relationships with plants and animals. And may microbes actually cause disease? This concept became known as the germ theory of disease. It was an important advance because originally people believed that when somebody became ill, it's because they breathed in poisonous vapors. Like they believed there were poisonous vapors in the swamp, and somebody walking through the swamp might later come down with cholera because of the poisonous vapors. In reality, somebody walking through the swamp will be exposed to cholera, and they may breathe it in and then get cholera. Any question about any of that? All right, about this time, well, a little earlier, there were people advocating that doctors should wash their hands. And Semmelweis, I may be mispronouncing his name because I'm not German, suggested that doctors, before helping a lady give birth to a baby, they should wash their hands. And he noticed that both postpartum and puerperal fever were reduced if the OB-GYN doctors washed their hands beforehand. And so he required that all of the doctors under his supervision, which he was in charge of the, what do you call that? The section of the hospital that gives birth to babies. I don't remember. But he required that of his doctors, and the president of the hospital was so insulted that he suggested doctors wash their hands. They actually fired Semmelweis. The concept of germ theory disease was then further advanced by Joseph Lister, who used chemical disinfectants to prevent wounds after surgery. And this dramatically reduced surgery-related infections. Previously, before Joseph Lister's day, whenever a surgeon operated, the patient almost always got an infection in the surgery wound, which really killed about 50% of the patients, the infection itself. And Joseph Lister showed that if you use chemicals to disinfect the surgery patients beforehand, as well as use the chemicals on your, like your knife and your scalpel, you can dramatically reduce the infection from a surgery wound. The germ theory of disease was then finally proved by Dr. Robert Koch, and this can be pronounced different ways, Coach Koch. The German pronunciation, I can't pronounce it because it's a sound that's an ex-guttural sound deep in your throat, and I'm not German, so I can't really say it. It's something like "Ugh." If anyone speaks German, you could say it for me, because like I said, I don't speak German and I can't make that sound. Anyways, he provided the first evidence of a bacterium causing a specific disease. What he did was he followed Bacillus anthracis and transferred the Bacillus anthracis to an organism, saw the Bacillus anthracis grow, and then saw the organism come down with the disease, anthrax. And this provided the first evidence of a germ causing disease. He didn't propose the theory, but he proved the theory. He then wrote up the steps that he used to prove that Bacillus anthracis causes the disease anthrax. And this was the steps that he suggested people use to show that a given organism causes a specific disease. And this became known as Koch's postulate. Any question about any of that? So first he wrote up this showing that Bacillus anthracis causes the disease anthrax, and then a few years later, he wrote up the steps he used, which became known as Koch's postulates. Around this time, we had our first vaccine. The first vaccine was made by Edward Jenner, and what he did was he made a vaccine against the disease smallpox. What he did was he inoculated a person with the cowpox virus. This is a virus that normally infects cows, but if you handle the cow, especially the cow with the cowpox, meaning the pox itself, you could become infected with cowpox. And he noted that people who, like milkmaids, who tended to get cowpox, were immune to the disease smallpox. And he never had any evidence other than, what he heard and what he observed. But he actually injected James Phipps and his own son with cowpox. And then when smallpox went through wherever they were living, I'm not sure where it was, a village in England or someplace in a city in England, I don't know. Those two boys did not come down with smallpox, showing that they were immune to the disease smallpox. Vaccine is a word, or vaccination, sorry, vaccine, same thing, from the word vaca, which is a word that stands for cow. And that comes from cowpox being used as the first vaccine against smallpox. Any questions about that? The protection one derives from a vaccine is called immunity, where a person becomes immune to a specific disease, in this case, smallpox. You understand at this time, nobody knew why or how the cowpox vaccine was working. All they knew was it helped prevent people from getting smallpox. So it was the first vaccine, but nobody knew how it worked, so nobody knew how to make another vaccine. Until the work of one person, guess who that was? Oh, where am I? Well, I'll just mention it was until the work of Louis Pasteur. He went and studied making vaccines and how you make vaccines and why they work. And he discovered that cowpox was a virus similar enough to smallpox that when the body responded with making antibodies against cowpox, at least some of those antibodies cross-reacted to smallpox because the virus was so similar to cowpox. Any question about any of that? All right. Around this time, we had the idea of chemotherapy, where if disease was caused by a microbe, maybe we could find a chemical that we could use to treat the disease. And Paul Ehrlich was actually the person promoting this, talking about a magic bullet, a chemical that would be applied to a person with disease, and the chemical would not harm the person, but it would kill the microbes causing disease. You should know a chemotherapeutic agent is any agent that can be used on an infectious disease. Nowadays, when we use the term chemotherapy, we almost always use it only with the therapy used against a cancer patient. But the term initially meant the treatment of any disease with any chemical, chemotherapy. We won't use it that way because common usage use it as a drug for treating cancer. So we will call an antibacterial agent for a drug used against a bacteria causing disease. And these antibacterial agents could be synthetic drugs or an antibiotic. An antibiotic is, by a textbook definition, meaning a chemical produced by a living organism, either a bacteria, an archaea, or a fungal organism, and then that agent inhibits or kills other microbes. By this textbook definition, penicillin V is a antibiotic because it's produced by another living microbe. But carboxylin is not an antibiotic because it was not made by a living organism. I believe, if I have my memory correct, carboxylin is one that is made in the lab, and everything is made in the lab for that molecule. There's something like a semi-synthetic, you don't really need to know this. So there's a antibiotic, meaning one that's made by a living organism, and then there's a synthetic, that's one that's man-made, and there's something like called semi-synthetic, and that's one that we make it in a lab by a living organism making it, such as penicillin V, And then we take that molecule, penicillin V, and we change it, usually adding parts to it, but you could remove parts or change the order of the parts or whatnot. The point is you're changing the molecule. And that we call man-made or at least semi-synthetic. And like I said, penicillin G, as well as penicillin V, are antibiotics, meaning a living organism makes it. But then we took that and we modified the molecule, and that's how we make, just drew a blank there. Ah, I had it on my tongue earlier too. Ampicillin, there we go. That's how ampicillin is made. It's first made by a living organism, and it makes penicillin G. And then we take that penicillin G and we apply other parts to it, other atoms to it, to get ampicillin. Any question about any of that? All right. The first chemotherapeutic agent, or the first antibacterial agent, was actually quinine from the bark of the cinchona or the cinchona tree. Both pronunciations are correct. And the bark of the cinchona tree was used to treat malaria. Does anyone have a guess when this bark of the cinchona tree was first used in Europe by the people against malaria? Anyone have a guess?
Are we talking about the Spanish conquistadors?
Possibly. So when's your guess? I don't know when the Spanish conquistadors were, so
Oh.
you're going to have to give me a year
In
here.
the 1600s?
That's correct. You're the first class who's ever gotten it correct. And that is Jesuit missionaries in Peru noticed the natives eating the bark of the cinchona tree to treat their malaria. And they noticed that the bark of the cinchona tree cured the patients of malaria. And so the missionaries sent the bark back to Europe, and it was first used in Europe in the mid 1600s to treat malaria. Who knows how long the natives of Peru were using the bark of the cinchona tree before the mid 1600s. So the first anti, well, let's say chemosynthetic agent, because malaria isn't a bacterial agent, chemosynthetic agent or antimicrobial agent was first used in Europe in the mid 1600s, long before people even knew about microbes. And then Paul Ehrlich went studying different drugs, and something like the 500th drug that he tested actually cured a rabbit of the disease syphilis in 1910. And he called this the magic bullet, a drug. It was an arsenic drug called salvarsan that was then later used to treat people for syphilis. Any question about that? All right. Just as a side, you don't need to know this for the exam, but just for your amusement. Before Paul Ehrlich found a drug, which was salvarsan, that could treat syphilis. If a patient had syphilis, the only way to treat it was to give the patient malaria and hope that the high temperature fever of malaria cured the patient of syphilis. And then they would use the bark of the cinchona tree to treat the patient and hopefully cure them of malaria. That was the only cure they had for syphilis before 1910. Any question about any of that? Obviously, that was a very dangerous procedure. You had to infect the patient with malaria. Hopefully, the patient didn't die of the combination of syphilis and malaria. And if the temperature was high enough the patient got from the malaria, it could cure the patient of syphilis. And then you could use the bark of the Sakono tree to cure the patient of malaria. Obviously, it worked in some cases, but in some patients, the patient obviously died. Or in some patients, the syphilis was not cured, as well as in some patients, the malaria was not cured. So there was risk. All right. The birth of modern chemotherapy, meaning really the birth of antibiotics, happened first with Paul Ehrlich in 1910, coming up with the drug, salvarsan, that could cure a patient of syphilis. In 1928, Alexander Fleming discovered the first antibiotic. What he did was he streaked out a bacteria on his plate, and he was a sloppy microbiologist, and he got fungal contaminant growing on the plate. And then for some reason, he allowed the plate, probably just forgot about it. He left the plate on the counter of his lab bench or his table, and both the bacteria and the fungi grew. If he had grown it in the incubator, the fungi would not have grown, at least not grown well, but the bacteria would have grown. Anyways, when he then went on like a weekend holiday, when he came back in his lab, he saw the plate there and he looked at it and he saw something like this where the bacteria were growing there and the fungi growing there. And instead of throwing it away because it had a contaminant on it, he looked and he looked carefully and he noticed that there's no bacteria growing around the mold colony. And he correctly concluded that something was being produced by this mold colony diffusing through the agar, and he called that something an antibiotic, which was later called penicillin. I'm not sure if it was Alexander Fleming who called it that or somebody else. It actually was penicillin G, for your information. He could not purify it any further than just getting a culture that he sent through a filter to remove living cells. And all it was filtered media containing penicillin. It's not something you can inject in a patient to cure them. So the first antibiotic that became available was actually a sulfur drug, sulfonamide, which was a man-made chemical. And so it was easier to separate out the antibiotic from the other molecules. And so sulfonamide, or the sulfur drug, became the first antibiotic that became clinically available for use. And it became available in 1935. Penicillin was not clinically tested until the early 1940s, meaning during World War II, and then mass produced. And if there's time, you guys should ask me the story about the first two cases of patients who were first treated with the antibiotic penicillin G being harvested from a mold. Any question about any of that? All right. Bacteria were once classified as plants, hence the term flora for microbes. You might hear me accidentally say microflora, because that's the term that I knew when I went through microbiology when I was an undergraduate many, many years ago. Microbiota is now the new term that has replaced microflora. So I should be using the word microbiota, just that that term wasn't around when I went through microbiology. And I'll try and remember to always use the term microbiota and never use the term microflora. And you can correct me if I ever use the wrong term. The normal microbiota are microbes that are normally present in and on the human body, and that we call the human normal microbiota. All animals and plants whose symbiotic microbial communities that are central to the animal and plant's welfare, meaning some of our normal microbiota really help us out. Any question about any of that? Oh, I forgot to mention, when we're talking about the normal microbiota, we're usually referring to the human normal microbiota. If we're talking about the normal microbiota on another animal, then you should state what animal. Like, for example, the normal microbiota on cats is a little different than the normal microbiota on humans. For example, both species would have Staphylococcus epidermidis, but only cats have the normal microbiota of Bartonella henselae. Bartonella henselae, if it gets on a patient, it will likely cause an infection. Any question about any of that? My point being, the normal microbiota of cats is a little different because cats normally have Bartonella henselae. Humans normally do not. And if they have it, it is usually causing a disease. Bartonella henselae, as far as we know, causes the kitty cat, no disease. The normal microbiota are normally found on or in the skin, the oral nasal cavities, the upper respiratory tract, the digestive tract, and the urethra, meaning the lower urinary tract. The normal microbiota are not usually found internally, such as in the blood, in internal organs and tissues, like inside your bone or inside your brain. The normal microbiota can prevent the growth of pathogens. They can compete for resources. I know you can't see my hand, but I've got Staphylococcus epidermidis right here on my hand. And if the Staphylococcus aureus were to fall there, it would be competing with the Staphylococcus epidermidis for space as well as for resources. And the Staphylococcus aureus may never get settled and it may not grow. So the normal pathogens can compete for resources as well as space, preventing the growth of a human pathogen. The normal microbiota can secrete toxins that inhibit the growth of a pathogen. And we briefly talked about one case of this where humans forgot the name of the organism, and normally causes the bacterial infection of the feet. And if you were to touch your boots or your shoes, which have the bacteria on it, and then touch it with your naked foot, the bacteria could transfer to your foot. And if it gets established, it could cause disease. But we could have other organisms around that inhibit the growth of the pathogen, that could be inhibiting it by simply competing for resources. And so the bacteria doesn't get established, or at least established well, and then eventually it'll die and go away. The normal microbiota can help out the host by producing nutrients and growth factors and then sharing those with the host. That is actually how humans get a significant portion of their folic acid and their vitamin K. The microbes in our gut can make folic acid and vitamin K. We cannot. And so if you were to eat like the pie I made from scratch, it would have nutrients in it, which could help the normal microbiota grow, but you don't want to have the nutrients around, like right before you inoculate the organism with what you're challenging it with. Some normal microbiota can also secrete toxins that inhibit the growth of a pathogen. We'll talk about this a little bit later, but E. coli, for example, can secrete a toxin that only inhibits the growth of Salmonella and not Serratia, what the heck is it? Shigella. So Salmonella and Shigella. It would not inhabit other normal microbes growing in your gut, like Enterococcus faecalis. And then the normal microbiota definitely helps us digest our food. E. coli is just one species in our gut that helps us digest our food so we can get more nutrients out of the zucchini bread. Any question about any of that? All right, the normal microbiota do not normally cause disease in a healthy patient. However, they can cause disease in certain circumstances, such as when the body's resistance is weakened, like the patient has AIDS, or the patient is taking chemotherapy for their cancer, and the chemotherapy weakens their immune system. Then, under these courses, the normal microbiota may cause disease. If the normal microbiota does cause disease, we call it a, oh gosh, drawing a blank here. Not doing real well here.
Sorry to interrupt. Is the class till 6:20?
It ends at 6:20. I was trying to remember what that is. We call it an opportunistic. There we go. It's an opportunistic pathogen. So it's not normally a pathogen, but it can be an opportunistic pathogen. I'm going by the clock on my computer, and it's now 6:21. So let me end it here and ask, are there any questions?
Will you have the slides available?
Not until the lesson is finished.
Okay.
All right. If there's no further questions, I'll end it here, and we'll see you possibly at 6:30 for the 6:30 lab. Bye.
Thank you.
You're welcome.